Hello, everyone. Thank you so much for joining us today for this webinar on applying in vitro phototoxicity assays. The webinar is co-organized by the Institute for In Vitro Sciences, or IIVS, and PETA Science Consortium International. I'm Bridget Rogers. I'm an advisor to the PETA Science Consortium, and I'm co-moderating today's webinar along with Christy Sullivan, who is the VP of Education and Outreach at IIVS. This webinar is part 2 in a 2-part series. So thank you if you joined us for part 1, which was last week on October 4th. And if you missed it, please watch the recording at the link that's shared in the chat. The link is the webinar title, so you click on that and it takes you to the video. The slides and a recording from today's webinar are also going to be posted at that same link later on today. Okay, so today we have 2 speakers who will be presenting case studies on phototoxicity test methods. First, we'll hear from Gretchen Ritacco from the Research Institute for Fragrance Materials, or RIFM, and then we will hear from Dr. Edward Chikwana from Corteva Agroscience. Everybody's on mute, but you can type any questions you have into the top questions toolbar. It's the little chat icon with a question mark. And in this software, the questions aren't going to be visible to everyone, but we will read them out when we get to the Q&A at the end of the second presentation. Please indicate if you have a question to a specific speaker, okay? All right, with that, we will go ahead and get started. Speaking first today is Gretchen Ritacco. Gretchen earned her Master of Science degree in Animal Science at Rutgers University. She has worked at the Research Institute for Fragrance Materials since 2009, where she is principal scientist. Gretchen leads the Photosafety program at RIFM, where her duties include evaluation of fragrance materials in support of RIFM's fragrance ingredient safety assessments and managing RIFM's photoallergy research. She is engaged in research collaborations with industry leaders to investigate new approach methodologies for evaluation of photoallergenic potential of fragrance materials. Gretchen is a member of the Society of Toxicology and both the European and American Societies of Photobiology. Welcome, Gretchen. Thank you for being here. I see that you're sharing and everything looks good. It's in presentation mode. So please go ahead.
Okay, great. Thank you, Bridget. And thanks to the PETA Science Consortium and IIVS for hosting this webinar series and for giving me the opportunity to talk a bit about my experience with photosafety testing. Okay, so to that end, today I will talk to you a bit about how RIFM conducts photosafety evaluations of fragrance materials. I'll also introduce you to our photoallergy research program and our collaborative efforts on new approach methodologies for photoallergy. So I'm going to start off by just providing some historical context. I'll move on to a description of our tiered approach to photoirritation evaluation with a case study. And I'll end my presentation by introducing you to our photoallergy research program, our ongoing collaborations, and why we believe that this effort is really critical to the fragrance industry. Okay, so RIFM has been the leading resource for the safe use of fragrance ingredients for over 55 years. Our evaluations have evolved over that period, from fragrance monographs to fragrance material reviews and group summaries to our current fragrance ingredient safety assessments, and from evaluation of discrete synthetic fragrance materials to natural complex substances. Photosafety has been an endpoint that we've been interested in since the late 1970s. And historically, photosafety has been an issue for fragrance materials, for cosmetics, and even for consumer products. If we think about lemons and limes, these are naturals that contain the most potent photoirritants, furocoumarins. There was an outbreak of photoallergic reactions in the 1960s. It turns out that TCSA and other salicyl anhalides were being used in detergents in the early 1960s. And that center picture shows an antibacterial soap ad from the 1960s, which contained hexachlorophene, which we now identify as a photoallergen. And then later on, musk ambrette. This was being used in men's aftershaves and colognes in the 1970s and maybe early 80s. It's a nitromusk. It's now known that it's a photoallergen. And the paper that described photoallergy to musk ambrette also described the phenomenon of persistent light reactors, or people who continue to react to sunlight even after they stop their use of the product containing the photoallergen. Now, if we broaden our horizons and consider pharma, in the United States, photosafety is one of the main adverse events that can cause a drug to be withdrawn between phase 1 and post-approval. And in fact, 34% of post-approval withdrawals are due to immunotoxicity and photosafety. Again, this is for pharma and the treatment of disease. And in those situations, assuming some risk is acceptable. Most consumer product companies, on the other hand, won't move forward with a material that has photosafety concerns because the assumption of that same risk really doesn't make sense when you're talking about consumer products. So when I talk about photosafety, at RIFM, we're interested specifically in photoirritation and photoallergy. These are two mostly distinct endpoints. There is some overlap, but I just want to be clear and start off by defining these terms. So photoirritation, this is analogous to skin irritation with UV exposure required for a reaction to occur. An example of a photoirritant would be furocoumarins. These are the most well-known photoirritants and also the most potent. And for materials that are identified as photoirritants, for fragrance materials identified as photoirritants, risk management can just be a simple concentration limit. So that would mitigate the risk to consumers. Photoallergy, this is analogous to skin sensitization. Again, UV exposure is required for the reaction to occur. And an example, as I already mentioned, is musk ambret. Historically, for the fragrance industry, risk management for a photoallergen has been an ingredient ban. Okay, so moving on to how we conduct photosafety evaluations. When we're assessing materials for photosafety, the very first thing that we want to know is whether that material absorbs UV or visible light. This tells us that the material has the potential to be photoactivated. If it doesn't absorb UV or visible light, it might still be a skin irritant and it might still be a sensitizer, but it's not going to be a photoirritant or a photoallergen. So we calculate absorbance on a molar basis. And then we compare that molar extinction coefficient, that value, to a benchmark value. This tells us whether the absorbance is considered significant or not. If there's no absorbance in the UV or visible light range, or if the absorbance demonstrated is insignificant, we can use that data alone to clear it for both photoirritation and for photoallergy. And the benchmark that we use comes from a paper out of Pfizer published by Henry, where they looked at 35 known phototoxic materials, and they found that all but 5 had molar absorbances above 2,500 liters per mole per centimeter. Most were actually greater than 3,000. So that benchmark of 1,000 liters per mole is pretty conservative. Okay, so now let's talk specifically about photoirritation. For materials that demonstrate significant absorbance, and in the absence of available data, there are 3 assays that we use at RIFM to address photoirritation. We test in a stepwise manner, and we start with a hazard-based assay, the 3T3 neutral red uptake. If a material is predicted to be a photoirritant in the 3T3, we would then move on to a risk-based assay. We'd move on to the reconstructed human epidermis phototoxicity assay, or RHE assay. Now at RIFM, if we derive a no effect level from an in vitro assay, we typically want to confirm that no effect level in a human test. So at RIFM, typically if we've done an RHE phototoxicity assay, we will also do a human photoirritation test. And I just want to be really clear about this. That human testing is done only to confirm no effect at the given concentrations. Okay, so let's move on to a case study. So we'll look at a specific fragrance material. It's a material called ethyl vanillin, which imparts an intense vanilla odor. So ethyl vanillin, we looked into it. We saw that it absorbs UV light. This indicates that it's got the potential for photoactivation. So you can see the UV absorbance spectrum up on the screen. You can see that within the range that we're interested in, between 290 and 700, there's an absorbance maximum probably around 310 nanometers. When we calculate that absorbance on a molar basis, we saw that it was actually, that MEC value was actually greater than 7,000, so it's above our benchmark. So our question is, okay, if, we've got the potential for photoactivation, we have a concern about the material, but if a person has this material on their skin and they go out in the sun, will they develop a photoirritant reaction? So as I mentioned, after UV absorbance, the 3T3 is the first step in our stepwise photoirritation testing paradigm. So this is a hazard-based assay. The test system is a monolayer of 3T3 fibroblast cells derived from mice. And in this assay, we're really just comparing cytotoxicity between the UV exposed cells and the non-UV exposed cells. We're assessing viability with neutral red vital dye. So again, we're comparing dose response curves of cells in the absence of UV light and those in the presence of UV light. And the difference in those dose response curves is called the mean photo effect, or MPE. So on the screen, we've got two trials with ethyl vanillin. Viability is on the y-axis and the concentration of test materials on the x-axis. And you can see that the UV exposed cells, those are the ones with the yellow blocks, there was a significant loss of viability as the test material concentration increased compared to the non-UV exposed cells. And in fact, the MPE for both trials was greater than 0.15, which according to the prediction model would classify this material as a photoirritant. So when you get into photosafety, some of the things you'll hear about the 3T3 and some notes I should kind of, make are that solubility can be an issue. So if you have a test material that has solubility issues, this maybe isn't the best assay for you. You'll also hear people say that the 3T3 is overly sensitive. I think you have to be careful with semantics here. I don't think it's so much that it's overly sensitive, it's just that you may be testing your material at concentrations that you're unlikely to see in real life. But if that 3T3 is negative, you can be confident that the material is really not going to present a concern for photoirritation. Okay, so if photoirritation is predicted by the 3T3, then we would move on to a reconstructed human epidermis phototoxicity test. So again, for ethyl vanillin, we saw both trials, it was predicted to be photoirritant with the 3T3. So then we moved on to the RHE. So now the RHE, the test system, is reconstructed human epidermis tissue. So it's a tissue model. It's human derived, it's got a functional stratum corneum, and it's a risk-based assay. So now we have to choose 3 concentrations of our test material to look at. So those concentrations are shown on the x-axis. We looked at 1%, 3%, and 6% ethyl vanillin. The UV exposed tissues are compared to the dark exposed. Viability is expressed as a percent, and that's on the y-axis. And in this graph, the UV exposed are the yellow bars and the dark exposed, the blue bars. So while you do see a dose-dependent decrease in viability, you'll notice that it's really not affected by UV exposure. At every concentration that we looked at, there's not really a huge difference between the UV exposed and the dark exposed tissues. The only significant difference that we did see in viability between UV exposed and dark exposed tissues was with our positive control chlorpromazine, and you can see that the UV exposed are much less viable than dark exposed at the same concentration. And that's as expected. That tells us that the assay is working as it should. And again, for this assay, a decrease in viability of 30% or more at any dose would be a positive. So this assay, a couple of notes about it. This assay is really nice for materials that have solubility issues. It can also be used for final formulations. We don't do that at RIFM, but I imagine consumer products companies probably do that. It's nice because you can do topical application of your test material. And the other really nice thing about it is that because it's a tissue model, it's less susceptible to UVB toxicity. So you can incorporate UVB irradiation into this assay. At RIFM, we've done that for materials that are predominantly UVB absorbers. Okay, so again, the no effect level for photoirritation found in that RHE test. At RIFM, we typically like to confirm that no effect level in human volunteers. So we typically conduct a human photoirritation test. Again, I want to be really crystal clear about this. This testing is done to confirm no effect. So we never test in people expecting to see reactions. The protocol that we use calls for 25 volunteers. We look at 3 concentrations of test material. We use the same 3 concentrations from the RHE as long as they were all negative. And then we also look at the vehicle alone. We looked at distilled water. Those are negative controls. And in this test, what we're doing really is comparing the irradiated test sites to both the non-irradiated test sites and blank test sites. Blank test sites would be sites that are irradiated, but they are not treated with test material. And the comparison between those 3 sites tells us whether photoirritant reactions are present or not. Let me see, some other notes about our test protocol for human testing. We use a combination of UVA and UVB irradiation to kind of mimic sun exposure. We do a 24-hour patch with test material. We remove the patch, then do the irradiation, and then the sites are scored 24, 48, and 72 hours after patching. So again, for ethyl vanillin, we looked at the same 3 concentrations, 1, 3, and 6%. And this human test confirmed a no effect level for photoirritation of 6%. We had no photoirritant reactions at irradiated or non-irradiated sites at any dose of ethyl vanillin. So in summary, ethyl vanillin does not present a concern for photoirritation at concentrations up to 6%. So, the background information on this testing, again, was that we had this physical chemical property, UV absorbance, that revealed a concern for photoactivation. We conducted in vitro hazard-based testing, that was the 3T3, and that test suggested that there was the potential for photoirritation. When we moved on to risk-based testing, the RHE and the human photoirritation test, we found a no effect level for photoirritation equal to the maximum concentration tested. So again, this tells us that ethyl vanillin is okay to use at concentrations up to 6%. So early in 2022, we had a manuscript published describing this tiered approach that we use for evaluation of photoirritation of fragrance materials. And that was published in Regulatory Tox and Pharmacology. This publication actually serves as both an update to RIFM's criteria document specifically for photoirritation, but it also provides data on 108 fragrance materials which demonstrated significant UV absorbance and went on to be tested for photoirritation using that tiered approach. And I should point out that this was actually a joint effort between RIFM and IIVS. Okay, so let's move on to photoallergy. I showed you this slide earlier and I want to touch on it again. So we can manage risk for materials that are identified as photoirritants, but historically, materials that are identified as photoallergens or fragrance materials have been banned. So for RIFM and for the fragrance industry, it's really critical that we're able to distinguish between these two activities. And while we do have many guideline studies to predict photoirritation, that's not the case for photoallergy. To address photoallergy, validated guideline study options are limited. UV absorbance tells us whether a material has the capacity to absorb light, and that would tell us whether it has the potential to be photoactivated. The reactive oxygen species, or ROS assay, if you tuned in last week, you got to hear a lot about this assay. That ROS assay tells us whether the material has the potential to be photoreactive. In all honesty, the ROS assay is new to RIFM. We haven't really used it yet, but we are investigating it, and it may be added to our repertoire of testing. So these are both in chemical assays. They can be used to tell us something about the likelihood of a material being a concern for photoallergy, but they're not specific to photoallergy. So if the results are negative, that's great. The assays can be used, or that data can be used to clear a material for photoallergy. But positives don't necessarily mean that the material is a photoallergen. But again, because materials identified as photoallergens can result in the ban of a fragrance material, finding new assays is really the focus of our research program. Okay, so, if we've got a material, it exhibits significant absorbance, there's no available data to complete the photoallergy assessment. Well, what do we do in cases like this? Well, thankfully, of the more than 2,000 fragrance materials that we've evaluated for photosafety, there are only about 100 that fall into this category. And again, really, those 100 materials, really the only mark against them is that they demonstrate significant absorbance of UV light. Through collaborations, we are determining if in chemical or in vitro assays for skin sensitization can be modified by adding a UV exposure step and then making the comparison between the UV exposed and the dark exposed to evaluate photoallergy. So we have a few collaborations ongoing, and the partners for these collaborations, we have one that is among RIFM, Shiseido, and IIVS, where we're looking at the photo direct peptide reactivity assay, or DPRA, the photokeratinescence assay, and the photo human cell line activation test. And some of that work, some of the work on the photokeratinescence was actually presented at SOT back in March. We've got another collaboration with a company called Sensigen. They're a Swedish company. They've got an assay for skin sensitization called Guard Skin Dose Response. And again, we're investigating a modification of that protocol to assess for photoallergy. And some of that work was presented at SOT in March as well. Those two collaborations, the Shiseido and IIVS and the Sensigen collaboration, those have been going on since the start of the pandemic, so since 2020. We just started a new collaboration with a company called Immunosearch. They have a skin sense assay called Census, and we're looking at the photo version of the Census assay. Okay, so the assays we're looking at, they're well established in the skin sensitization space. Each addresses a specific key event in the adverse outcome pathway. That's shown in this graphic. Kind of looking forward and based on some of the results we've gotten so far, we anticipate that it's probably unlikely that we'll have a standalone assay for photoallergy. More likely, we'll use a combination of assays to provide us with information on the likelihood of fragrance materials presenting a concern for photoallergy. And all of these collaborations, the first thing we do is kind of determine fit for purpose. We do this by testing known photoirritants and known photoallergens. We do recognize that there is some overlapping activity. So we've tried to kind of categorize reference or proficiency chemicals as predominantly photoirritant or predominantly photoallergenic. And in a few cases, we had materials that really seemed to demonstrate both activities equally. Frankly, this has probably been the biggest challenge with these research collaborations. As you can imagine, it's really difficult to assess an assay without a large set of reference or proficiency chemicals. And it really doesn't seem like there's a large set of known photoallergens. So that's been a real challenge. But in summary, it's through our tiered testing approach that RIFM can effectively address photoirritation. We do that by looking at UV absorbance, the 3T3, and the reconstructed human epidermis phototoxicity assays. And then, as I mentioned, we typically confirm that no effect level with a human test. And it's through collaborations with research partners that RIFM is working towards new approach methodologies for photoallergy. And again, as I mentioned, we've got 3 collaborations going on currently, one with IIVS and Shiseido, one with Sensigen, and a new one with Immunosearch. And at this point, I'd really like to thank you for your attention.
Wonderful. Thank you so much, Gretchen. That was a great presentation. Okay, and a reminder, we're going to hold any questions you have until the end of the next presentation, which we'll go ahead with. But please go ahead and submit any questions you have into the toolbar, okay? All right, speaking next will be Dr. Edward Chikwana. Edward earned his PhD in environmental chemistry from Portland State University in 2006 and an MPH with a concentration in environmental health from Indiana University in 2016. He currently works at Corteva Agriscience as a global human health risk assessor within the Crop Protection Regulatory Sciences function, where he is a subject matter expert in risk-based dietary and non-dietary human exposure assessments to support registration of plant protection products across various geographies. Before joining Corteva in 2018, he was a tenured professor of chemistry at Franklin College of Indiana. Edward currently teaches as an adjunct professor at the Indiana University Richard M. Fairbanks School of Public Health, where he is the primary instructor for 2 graduate courses, Fundamentals of Toxicology and Human Health Risk Assessment. Edward, thank you so much for being here. I see that you are presenting in presentation mode, so it looks good, and feel free to go ahead.
Thank you so much. And good morning, good afternoon, good evening, wherever you are. My presentation is based on a new approach methodologies framework that we are proposing to do phototoxicity risk assessment for plant protection products or agrochemicals. This work is based on the publication presented here. So you can use the QR code to access it as needed if you want a detailed explanation of all the work that I'll summarize in this presentation. So throughout this presentation, I will give a high-level introduction to EU current guidance on phototoxicity testing for agrochemicals, and then walk you through our proposed framework that uses a risk assessment approach, and then apply this framework to a case study that we also included in the publication. So currently, the European Food Safety Authority, or EFSA, is using an old trigger value of 10 per molar per centimeter molar extinction coefficient to trigger photosafety assessment. And in our approach, we are proposing moving to the updated OECD value of 1,000. And interestingly, there is detailed guidance on how to test or to do hazard characterization, but it's not clear what to do after you have identified that your chemical is phototoxic. So what we are proposing is a framework that allows you to be able to use current EFSA guidance for risk assessment of plant protection products to do a risk assessment for phototoxicity. And in our approach, we're also proposing using a benchmark dose approach to deriving a reference value based on in vitro studies. And then different agencies have models for estimating exposure, but since this approach was targeted to the EU, we are also using the EFSA model to calculate or estimate the potential exposure to agrochemicals. And then I'll walk through all this using a case study. So in a risk assessment approach, once you've identified a hazard, it is important for you to also account for exposure, and then a combination of the hazard and exposure gives you the risk or the likelihood that an adverse effect will occur. So I typically use an example of lions, which we all know present serious hazards to our health, but we still go to the zoo or on safaris because they are appropriate mitigation measures that allow us to interact with the lions without significant health risks. And that's the same with using plant protection products. Just because you have identified a hazard, it should not end there. You want to make sure that the exposure is also significant enough for there to be a risk. And in most cases, there is a product label that provides risk mitigation to that effect. So coming from this approach, we're looking at current EU guidance. And what the current guidance does is it does provide you with all the tests that you can do. And I will not explain each of the tests because Gretchen did a much better job than I would have done explaining each of these tests. But what the current EU guidance walks you through is how to test for phototoxicity and the different levels that you can test. But it doesn't say what should you do once you have a positive test. And this is the same as just identifying the hazard and then leaving it there. In the guidance, however, there is a statement that says a positive result shall be taken into account when considering potential human exposure, but then it does not say how to consider the human exposure. So our framework is trying to bridge this gap and provide an approach that is consistent with how EFSA currently evaluates all agrochemicals. So in our approach, what we are doing is we are proposing to update the trigger value for the molar extinction coefficient to use 1,000 as is consistent with current OECD guidance. And also we are adding this risk assessment approach in a tiered fashion where initially you can do a first tier or tier 0 approach where you can use the different OECD guidance to develop an in vitro based reference dose, use the EFSA models to calculate what your exposure is, either using default demo absorption values or generate your own product specific demo penetration based on the application parameters of your product. And then if all else fails, then that's where risk is identified. And currently, there is no guidance for in vivo assays for agrochemicals, although we looked at different in vivo tests that are used for pharmaceuticals, but none of those are validated enough for them to apply to agrochemicals. So at the moment, we are only going to focus on using the available in vitro assays to provide a reference concentration. So I'm going to use our proposed framework, which is a 5-step approach, and walk you through an example of a case study using a nematicide. We had 2 different case studies in the publication, but I'm only going to represent one in this. So the first step would be to use the updated guidance to see if your product is phototoxic or not by comparing your molar extinction coefficient to a trigger value of 1,000. And then once you have that, then you can do the phototoxicity assays to be able to quantify your reference concentration. So initially we are proposing starting with the OECD 432, which is based on a bidimensional mouse cell line. And for this cell line, you need to have a 20% decrease in viability between the exposed and the non-exposed, and then that tells you whether or that tells you the concentration at which your compound becomes phototoxic. So in our case study, we had two replicates, and what we are proposing is to not just pick a value where you see this difference in cytotoxicity, but to use a benchmark dose. And the EFSA uses a tool called PROST. So we were able to model our value and determined a lower limit concentration and an upper limit. And since the lower limit is more conservative, we proposed using that value. And based on the volume that you apply in your 96 well plate and the surface area, we're able to come up with a conversion to convert the concentration from micrograms per mil to microgram per square centimeter, because in the model that we use to estimate exposure, the exposure is given in micrograms per square centimeter. So once you have calculated your reference concentration, you can then use the available models to calculate the expected demo exposure. So in exposure models for agrochemicals, there are different scenarios that are considered, mostly mixing and loading of the concentrate, and then application of the diluted spray solution. And then we are also looking at potential exposure to bystanders or residents through spray drift. So these are the scenarios that we consider relevant for acute exposure. Other scenarios that could be considered could be oral exposure, but looking at how the models calculate, we were able to show that any contribution from the oral route that might end up moving to the skin is very minimal compared to exposure during application. So when you add those values, the contribution was very minimal. So we are only going to consider these three scenarios. And then in performing the risk assessment, we looked at what would be the risk without any PPE, and for the most part, exposure to agrochemicals for professional operators is assessed here. Assumes that you're wearing shoes, socks, long pants, long-sleeved shirts, but no additional PPE. So based on that, we were able to show that for our case study, the total exposure was 72% of the reference dose. But when you included gloves, which are typically worn or recommended on product levels due to other hazard categorization, that risk reduces to 6%. And then when you look at bystander and residents who are exposed through spray drift of the very dilute solution, you can see that the exposure is even much less at 0.1% for a child and 0.3% for an adult. So after we had done this test, and after we had publicized this work, we had planned to do an OECD 498 test using the 3D reconstructed human skin model. And what this graph shows is on the y-axis is your estimate of toxicity, and then on the x-axis is your estimate of exposure. And you can see that anything above the yellow line would be high, potentially risk of concern, and then anything below the yellow line indicates no or low risk of human concern. And you can see when you go from the OECD 432 to the OECD 439, you can show that the risk is even much lower. So if you are doing these assays and you do have data for both the 432 and the 498, we are proposing to use the 498 as this test system may be considered biologically more relevant since it's based on a human cell line. And in conclusion, what our proposed framework is doing is, in addition to the hazard ID that is identified in EFSA guidance, is to update the trigger value to be consistent with current OECD guidance, and also to be able to determine a reference concentration from in vitro studies. And then we are adding a risk assessment approach where exposure characterization is going to be based on whatever current model is available for estimating demo exposure. And this can potentially be refined by doing a product-specific in vitro demo penetration refinement. And with those pieces of information, you'll be able to properly characterize the risk from phototoxicity. And with that, I thank you for your time and wait for any questions that you might have.
Well, thank you very much, Dr. Chikwana, for your presentation. Hey, everyone, I'm Christy Sullivan from IIVS, and we'll go through the Q&A now. If you would please, if you have any questions for either of the speakers, put them into the question module and we'll go through them one by one. So right now we have a couple of questions for Gretchen, but I'm sure more will come in. Let's see. So the first question is how frequent, but I'm also wondering, is it frequent that human testing confirms a NOAL, and are that data published, those data?
Sure. So if we're doing an RHE, we're almost always doing confirming the no effect level that we derive from the RHE in humans.
Okay.
The only times we have not confirmed the no effect level in humans are when we've already got data on a very similar, structurally similar material. So, you know, I presented about ethyl vanillin. Vanillin was also kind of went through that tiered testing approach, but we didn't do the human test because we already had data on ethyl vanillin. So that would be a case where we wouldn't, you know, confirm that no effect level in humans. And really the only other situation I can think of, I don't think this has happened with us yet, but I guess if we tested a material in the RHE at levels so much higher than the use level, then maybe we would opt not to confirm a no effect level in humans. That's the only other situation I can think of, but it would really need to be a lot higher, like probably more than 25 times higher than use levels. And as far as the data being published, all of our fragrance ingredient safety assessments are available for free. We have the Fragrance Material Safety Resource Center on Elsevier. It's fragrancematerialsafetyresource.elsevier.com.
Okay. Fragrance, say that again so people can write it.
Sorry. It's Fragrance Material Safety Resource, all one word, .elsevier.com.
Excellent. Okay, thank you. Also for you, Dr. Ritacco, in the RHE phototox test, are the tested concentrations directly comparable with the concentration that a user will be exposed to? And I guess you already covered a little bit of this. Or would the NAM assays be enough evidence for hazard assessment in a regulatory context?
Okay, so when we're doing the RHE, we do have access to use levels in finished products. So we try to choose concentrations around those use levels. We try to choose a concentration above use levels and a concentration below use levels, so that, so we can, kind of assess what would happen to a consumer in real life. We, as far as is the human testing necessary, probably, I have a feeling that it probably isn't always necessary. We have never seen, if anything, we've seen more sensitivity in the RHE than in people. We've never seen a reaction in people after testing the same concentrations in the RHE. So I suspect that the RHE would be enough, but I can't really say in terms of a regulatory context whether it would definitely be enough.
Yeah. So Dr. Chikwana, we have a question for you. You mentioned potential refinement using product-specific dermal penetration. However, was the potential for phototoxicity to affect or increase dermal penetration considered when proposing the framework? And would in vitro dermal penetration studies need to be conducted for under simulated sunlight conditions?
That's a very good question. And in the projects that we do, we currently have guidance for normal dermal penetration studies for all other ag uses, and they are not
Right.
specific to phototoxicity testing. So we did not consider the possibility that irradiation might reduce or decrease the dermal penetration. So that's something that maybe we could consider, but for now, based on the guidance, and especially if you end up using the EFSA default dermal absorption values, they are very conservative compared to what we typically see when we do studies, that any increase that you might have potentially could be covered by the conservative nature of the models themselves. But that's a very valid point.
Great. Gretchen, did you have anything to add? Okay.
No, I didn't.
So this question maybe could be for both of you. So I will start with you, Edward. Is there a method to test mixtures for phototoxicity?
Yes. So for our product, so if you are making a product that you're going to put on the market that is a mixture of two active ingredients, you are required to do an acute tox spec for what you're actually selling, and that's the one that you also do a demo absorption on. So all the data that you are generating for the phototoxicity assessment is on the product rather than the AI. So whatever co-formulants you have, whatever other mixing partners you have, all that are covered in the value that you get. So it's a product specific test rather than an active ingredient test.
Great. Thank you. And Gretchen, did you want to add to that?
Sure. This is something that we are interested in because some of our fragrance materials are natural complex substances, so they're mixtures. We have tested, we have put some of these NCS materials in UV absorbance and in the 3T3 and in the 3D skin, the RHE. I don't think any are validated for that. When we do UV absorbance, it gives us a sense of how much it absorbs, but we can't quantify it because we don't have molecular weight defined for the NCS. But it does give us kind of a yes/no answer as to whether there's absorbance. We have done the 3T3. As I mentioned, it's not validated for mixtures. I think the 3D skin, I'm not sure that it's been validated, but it can be used for final formulation. So that might be like the best choice to look at mixtures for.
Mm. yeah, I think it would overcome some of the challenges with mixtures
Yeah,
as well. Okay, we have another question for both of you. So we'll start with Edward again. If the UV spectra suggested a UVB absorber only, would you go directly to the RHE assay and omit the 3T3, as this does not assess the light source?
So not necessarily. So my understanding of how the the two tests work is UVA is 95%, I think, of what you are dealing with. So we typically don't worry about UVB as much because when you're exposed to UVB, there's like a tolerance limit because you know when you go tanning, right? There is a limit to that. So in terms of penetration, we are not worried as much about UVB than we are of UVA. So essentially, what the tests are trying to quantify is mostly your UVA exposure.
Okay.
Maybe Gretchen would want to add more to this.
sure. Yeah, we had a few fragrance materials that were primarily UVB absorbers. To be honest, it wasn't something that I paid enough attention to. So I had put them through the 3T3. When they kind of were, we have an expert panel that kind of reviews all of our testing and our conclusions. So one of the experts on our panel suggested that maybe the 3T3 wasn't the most appropriate test to do, and so for those materials, we're now putting them into the RHE.
Okay. All right. This is again for both of you. Maybe this time we'll start with Gretchen. So regarding photoallergy, could you please expand on the use or potential use of sensitization potency for risk assessment? And are you planning to look into this after the hazard assessment tier approach is finalized?
So we kind of have everything on the table for photoallergy. Like I said, we don't have anything currently that we can use other than we are using UV absorbents. So if it doesn't absorb, we don't have to worry about photoallergy. We're starting to look into the ROS assay. For those skin sensitization assays, I don't know that potency will be something that I think at first it might kind of be more of a yes no kind of question. Photoallergy is tricky because it's not just the test material; it's the amount of sun, and so there's a lot a lot of variables. So I think that potency isn't something that we're so much thinking about right now. Right now, I think it's more of just the yes no kind of.
Okay. Edward,
So
did you?
for this project, we were not looking at that. But there is guidance that we do follow for plant protection products that are characterized as skin sensitizers. And the way that we currently look at skin sensitizer is when you are doing the test, you are testing the concentrated product. So this is the product that you buy from the store, and then that's the one that you are doing a yes or no. Is it a skin sensitizer or not? But there is guidance there that if it's a skin sensitizer, you also have to show that the diluted product, which is what residents might be exposed to through drift, is also not a skin sensitizer because the operator or the person who's mixing and loading can wear PPE, so you're not as worried about them as the person who might be exposed to drift. So we do need to show that the diluted product is also not a skin sensitizer. And there is work that is ongoing through CropLife Europe to come up with a risk assessment approach as well, similar to what we're doing for phototoxicity. But there is nothing yet that has been published by the regulatory agencies on how to do the risk assessment.
Okay. You're almost getting towards an answer to another question that was posed for you, actually, which was, would the risk-based refinement be dependent on a specific application scenario of the product?
Yes, that is correct. So it also might, when you look at what kind of product, is it an herbicide, is it a fungicide, is it an insecticide, how you apply it also affect how much might drift. So for example, when you're applying a herbicide, you're typically applying it downwards because you want to kill the weeds. So you typically don't expect a lot of drift. Whereas when you are doing a fungicide, maybe to an orchard, right, you are doing an airblast sprayer, so there is higher. So when you're looking at refinements, they will depend on what kind of insect active ingredient or plant protection product is, and how is it applied, at what stage of growth it's applied. So all that is factored into the model that calculates demo exposure. So
Okay.
that part should be aligned with the use patterns of the product as indicated in the models that we use to model the exposure.
Okay. That's really helpful. So we have one more question, and it's not directed to any one of you. It might be a challenge, so we'll see. We'll start with you, Gretchen. Is it possible to assess a potential increase of relative concentrations, for example, by bioaccumulation after repeated exposure? I don't know if there's a repeated exposure scenario.
So this is a challenging one. Yeah. We do consider aggregate exposure, so exposure from one fragrance ingredient that might be present in multiple product categories that are applied, that a single person might use, all of those products. Maybe they like that fragrance, they like fragrance in general, they use a lot of, so we do consider that, but I don't know that really answers the bioaccumulation aspect of the question.
Yeah. Edward, maybe
Yeah.
anything to add?
So I think similar to what Gretchen is saying, the way that you typically apply pesticide is different though to fragrances, right? For fragrance, you are intentionally choosing a specific brand, so you might apply the same every day. But for pesticide, usually you apply once per season, or you apply, if it's 3 times, it will say maybe over 7 days. So we typically are only looking at acute exposure on the day that you apply, because that's when you're exposed. So if it's a once per season, we are not worried as much about the bioaccumulation, because by the time you apply it again, um, any amount theoretically should be already gone. We do other tests as part of the tox package that we submit to show, the ADME processes for that product, and one of those is to show that your product is not accumulating in animals. So I would say that might be covered by other tox studies that we submit, but we are not looking at that from a phototoxicity perspective, similar to what Gretchen said.
Yeah. Okay. Well, there's always more improvement to be made, right? And everything that we're doing, all the testing that we're working on, and strategies for safety assessment. So we have come to the end of our questions exactly on time, which is some kind of magic today. I want to say thanks to all of the attendees for coming and listening. Many thanks to Drs. Chikwana and Ritacco. It's Ritacco, right?
Ritacco.
And Ritacco. Okay. Well, I messed up. Sorry. For sharing your expertise with us. I learned quite a lot in just a short period of time. So thank you very much for presenting and for answering these questions. Thanks to the PETA Science Consortium International for hosting this webinar with us. And just to remind you, the recordings of both the first and the second webinar are going to be on the PETA Science Consortium website together with some other resources. Thank you all and have a wonderful rest of your day.
Thank you.